Hello student. On the behalf of KTHM College Nasik and Department of Chemistry of this college, I am Dr. R.K.Pawar, Assistant Professor in Chemistry, Department of Chemistry in KTHM College, welcomes you in this M.Sc. Second Year Inorganic Chemistry class. Dear student, in previous lecture we discussed the metalloenzyme of zinc, that is carbonic anhydrase and carboxypeptidase. Now in this lecture we are going to discuss the next metalloenzyme of zinc, that is a zinc fingers. The name itself indicates the finger-like appearance of zinc, in which the zinc is coordinated, two or more zincs are coordinated in the structure. So let's discuss what is meant by the zinc fingers. Actually, small protein structural motif that is characterized by coordination of one or more zinc ion in order to stabilize the fold. Originally coined from the name that is a Xenopus laevius. Zinc finger. Xenopus laevius. This Xenopus laevius transcription factor third. So this is actually Xenopus laevius is a African toad species. From that the structure of zinc finger are deduced. So what is meant by this zinc motifs? So we know that the peptides are made up from amino acid. Peptide is folded. Folding is most important for the structural perspective of proteins. Now this folding is of different types, like alpha helix with alpha helix, alpha helix with alpha helix, beta sheet with beta sheet, secondary structure. So this alpha helix and beta sheet with beta sheet are the secondary structure. These secondary structures are furtherly rearranges between themselves is known as a tertiary structure. Tertiary structure is belonging to different kind of structural protein and structural motifs. Structural motifs means there is interaction between alpha sheet with alpha sheet, beta sheet with beta sheet, and so on. So this is known as a motifs. So zinc finger is shortly known as ZNF, where zinc and F is a finger. Now zinc finger is also called as a zinc motifs, and we already discussed this motif. So here now let's discuss the structure of zinc finger. Structure of zinc finger. We discuss. Now this is a structure of zinc finger. In that we know that here zinc is coordinated at the active site. This is a alpha helix and beta sheet structure, which are antiparallel alpha helix and antiparallel beta sheet structures. Now zinc is coordinated with the 2 histidine and 2 cysteine residues here. Now in that structure we can say that zinc is coordinated with two cysteine and 2 histidine residues. Zinc motifs is present in zinc motif. Zinc finger is also called as a zinc motifs, and what is meant by motifs we already discussed it. Now zinc motifs is coordinated to alpha helix and antiparallel beta sheets. So these are the structures of protein. In that zinc is again coordinated to two cysteine and two histidine group. Here, if you see the geometry is looks like a square planar. So it is a pseudo-square planar geometry in that zinc finger. It is not conformably deduced. So here, these are the structure of zinc fingers. Now, let's discuss the classification of zinc fingers or the classes of zinc finger. Actually, zinc finger is classified into various families, but it mainly classified on the basis of fold group domains. Now, this fold group domains is nothing but the structure, overall protein structure or the protein backbone, depending upon the that it divided into or it classified into three types. That is, first classification is that is, we know that the cysteine 2 and histidine 2, that is commonly known as the zinc finger. Then second, that is a treble clef, and third one is a zinc ribbon. Now these three classes are depending upon the folding domain, and this folding domain is depending upon the overall backbone of protein structure, and depending upon this structure, this zinc finger is classified into these three classes. That is, cysteine 2, histidine 2 is commonly known as a zinc finger. Then second one is a triplet clef, and third is a zinc ribbon. We are going to discuss how these fold groups are attached to the ligand placement. Now let's discuss. We are going to discuss the fold groups, and we know that our fold groups is zinc finger, treble clef, and zinc ribbon. So representative structure of that folding group is ligand placement. First, that is cysteine 2 and histidine 2. The structure of we know that the zinc fingers, the structure of this zinc finger is like that, because cysteine 2 and histidine 2 residues are present in the zinc finger. So the representative structure of that zinc is like that. Now the in ligand placement, this zinc finger is from two ligands. We know that in zinc finger there is a four ligand, two cysteine and two histidine. So that two ligands from nucleus and two from C terminus of helix. Now two ligand from nucle. Now what is meant by the nucle? So nucle is nothing but, the word nucle means the bone of hand which join the finger and palm is known as a nucle. So here if you see the zinc is coordinated at an active site, and here two histidine and two cysteines are joined, means two ligands are from nucleus and two ligands are from C terminus. This is a nucleus and this is a C terminus of the helix. So that's why this ligand placement is from two ligands from nucleus and two ligands from the C terminus of helix. Now the second, that is a triple clef. There is no any special representative structure of treble clef, but the ligand placement is that two ligand from nucleus and two from N terminus of helix or alpha helix. Here N terminus, we know that the each chain has a two terminus, N terminus and C terminus. Here our zinc finger is from C terminus and two ligands of treble clef ligand is from N terminus of the helix. Now the next one, that is a zinc ribbon. Zinc ribbon also have a same structure like that. It is a 3D structure. In that zinc is attaches two or more zinc ion are present actually. So this is a representative structure of zinc ribbon. Now what is about the ligand placement? Actually these two ligand each from two nucleus or nucleus. Means here from two nucleus. So these are the representative structure of folding groups and ligand placement. Now let's discuss in detail the structure of these folding groups. Classes. First we are going to discuss the zinc finger or the cysteine 2 and histidine 2 residues. The first class of histidine 2 residue is also known as a zinc finger. It is a, you know that the zinc finger is, this whole group is a zinc finger which have common. Zinc finger is a common mammalian transcription factor This folding groups is adopt, or this fold group adopts beta alpha fold and amino acid sequence. So in that class, or in cysteine 2 and histidine 2, the best example of this cysteine 2 and histidine 2 is a zinc finger. Zinc finger is a very common mammalian transcription factor. Now this fold adopts beta alpha and amino acid sequence, and this sequence is as like, that is X2 cysteine X, means here there is a 2 cysteine and 2 histidine residues. Again here histidine sequentially or alternately it will be attached to the cysteine, 2 cysteine and 2 histidine. So these are the general sequence. This is sequence in cysteine 2 and histidine 2 fold. This sequence is present in cysteine 2 and histidine 2 fold group. So these are the structure of this folding group of cysteine 2 and histidine 2. Let's discuss the structure of treble clef. Treble clef. Now in treble clef, treble clef motifs have a beta hair pin at N terminus and alpha hairpin at C terminus of peptide chain or the alpha helix. So this two ligand, now again this two ligands are bind to zinc ions in structure. This treble clef is the best example characterized by this treble clef. Zinc fingers are the nuclear hormone receptor. The nuclear hormone receptor is a best characterized zinc finger, treble clef zinc finger motifs. So this is a example of nuclear hormone receptor, is an example of this treble clef zinc finger motifs. Now let's discuss the zinc ribbon structure. We know that the zinc ribbon is from two ligands, each from two nucleus. So it will attaches the two ligands from two nucleus. So the structure of zinc finger is zinc ribbon. We know that the zinc ribbon, the ligand placement is like the two ligand each from two nucleus. So here the two in zinc ribbon, two beta hairpin from or forming two zinc finger, in zinc ribbon, two beta hairpin forming two structurally similar zinc binding subunits. So in zinc ribbon, the two beta hairpin forming two structurally similar zinc binding subunits. So here these are the structure of this zinc ribbon. Now in next we are going to discuss the application of zinc fingers. So the application of zinc fingers is that we know that the zinc finger is a bind to DNA and RNA for holding the structure or stabilize the structure. So the characterization of or the characteristics of characteristics of zinc ribbon or zinc fingers. Zinc finger is a 3D structural zinc motifs. 3D zinc finger is a is a 3D structural zinc motifs. Then second, it will bind. It binds to cysteine, and two histidine. Groups. Then third, that is, it will bind to DNA and RNA. It binds to DNA and RNA. Holding the structure together. Now, fourth application is that it will stabilize the fold. It stabilizes the fold. Then fifth application is that it will, or the zinc finger have a 30 amino acid and beta alpha sheet structure. And the sixth one, the sixth characteristics is that, I will write here, zinc finger is binds to plate within DNA substrate. So these are the characteristics of zinc finger or zinc motifs. Now let's discuss what are the main application of the zinc finger. Now let's discuss the applications of zinc finger. Zinc fingers. We know that the zinc fingers is structural motifs. It contains the zinc motifs. Now zinc is a very important and vital functions in many biological processes. It is main factor in DNA and RNA binding processes. So now let's discuss the applications of zinc finger. The first application of zinc finger is that zinc finger recognize zinc fingers helps in or function in DNA recognition. Then second, as we know that the zinc finger is bound to DNA and RNA to stabilize the fold. Due to zinc ion is at a center and active site, the folds are stabilized. So the second function is RNA packaging. Again, the next function or the next application of zinc finger is zinc finger helps in lipid binding. Lipid binding. We know that the lipid binding is very important for the digestion of proteins. So here, zinc finger is also helps in a lipid binding. Then fourth application is that it can also handle or helps in protein folding and assembly. In protein folding and assembly, zinc fingers play a vital role. Then fifth application is that it will be activate the transcription process. So in fifth application we can write as that it can activate or it helps in transcription activation. So these are the various applications of zinc finger. Instead of that, there are various biological and chemical processes in which the zinc finger play a very important and vital role. Now, in this lecture, we are discuss what is the structure, what are the classes depending upon the fold groups, and this fold group structures is depend upon the protein backbone. So, in that we discuss what is the structure of zinc finger, treble clef, and zinc ribbon. Now, recently we discussed the application of zinc finger. What are the characteristics of zinc finger? So, we will discuss the assignment. Now, the first assignment of this topic is write a short note on zinc finger. Second, what are the characteristics of zinc finger? Now, the third assignment is discuss the applications of zinc finger. So, these are the questions for assignment on our today's topic, that is zinc finger. Now, we will stop here. In next lecture, we are going to discuss the next metal enzyme of copper. Thank you.